This is a production of Cornell University.
All right, well, why don't we get started, guys? And I want to finish off this lecture because I want to start in on the next lecture today. I'm not sure I'm going to get done, but I want to finish off the sort of the safari that we're on, so the first lecture of soil ecology. And we were on this last of the classification schemes. Again, why you look at, or the question that you have is going to direct what you're looking at. Okay? Or another way of saying it, how you look at something sort of directs what you're asking, the kinds of questions that you're asking. Okay? So one of the classification things, one of the ways that we look at stuff, how we classify, is basically SR16S rRNA sequencing. It's basically, we're looking at literally the genes. Okay? We're looking at the RNA, not necessarily the DNA. We're looking at the RNA, but we're looking at a relatively conserved component of our body that we can watch it change over time. Okay? And that sequencing basically has broken up life into sort of three components: eukaryotes, eubacteria, and archaebacteria. And I just wanted to sort of walk us through the diversity that's here and to a certain extent its function. So you give guys, in essence, a safari. So eukaryotes are a large group. They include plants, fungi, animals, the ciliates, the flagellates, as well as the slime molds. We often have a hard time realizing that we're related to slime molds in a sense. Okay? When we talk about this, we often think of sort of the non-animal groups. So we're looking at the plants, from large to small plants, algae, fungi, the slime molds, and some protozoa. When we talk about animals, we're looking at small, again, large to small, elephants and gophers, but we're really talking about soil organisms here: gophers, moles, prairie dog, earthworms, slugs, all the way down to the nematodes. Okay? Generally, the things that you would think of as plants or animals fall into this group. Okay? From a plant sense, plants are kind of interesting. I've been sort of making this case that all this stuff is happening because the bacterial populations are driving nutrient cycling, but the reality is that this is a two-way street. And plants are a really good example of how these systems work. Okay? Plants, obviously they're photosynthesizing, they're taking carbon, inorganic carbon, and turning it into organic form. Okay? But they're doing more than that. Okay? They're basically supporting I guess it's the same thing. I mean, they're basically supporting an ecosystem. Okay? And the ecosystem, when we're talking about soils, a lot of that ecosystem is basically existing in the rhizosphere. The rhizosphere is the root zone. Okay? The root sphere. Okay? Now, here are these plants photosynthesizing, light's coming in, they're photosynthesizing, they're putting biomass out, and in their root system, they're basically putting exudates out. Now, these exudates can be doing this intentionally because they're trying to harvest something, okay? But it also could be unintentionally, where the plants are sloughing off skin, like we slough off skin cells, the plants are doing the same thing, okay? Well, that material that they are exudating basically becomes a food stock or a nutrition stock for organisms that are around, that live in this root zone. Now, it turns out if you look at the soil as a whole, the highest concentrations of organisms in the soil are basically sitting in these rhizospheres. They're sitting in this zone that is being influenced by plants. Okay? Now, these exudates sort of stimulate fungi as well as bacterial growth, and often will have 10 to 100 more microbes than in the bulk soil. Okay? Now, the relationship with these roots host harmless as well as parasitic as well as symbiotic organisms. So the soil organisms, the soil animals that you'll see out there are the nematodes, insects, slugs, and earthworms. We're going to be seeing, in the next couple weeks, we're going to be seeing a lot of earthworms. We're going to be seeing a number of nematodes, but a lot of different types of insects that are living in this soil environment. Okay? These are all basically heterotrophic organisms. There are some exceptions. With soil animals, they're all basically heterotrophic. They're all aerobic, mobile, mostly in topsoil and litter. There's few in compacted and very wet soils. The few is because they're aerobic. The protozoa and algae are very active and abundant in these wet soils. Okay? The algae basically are single-celled phototrophs. Okay, so they're not the heterotrophs that we think of as the soil animals. The protozoa are non-photosynthetic, but they are also single-celled, but they are organotrophs. Okay, this population is very mobile, and they prey very much on that bacterial population. Okay, so these guys are sort of those predatory. They're on the trophic scale. They're the predators. Fungi is another large group of this. They're active and abundant in most soils. The soils normally have to be moist and aerated for these fungal populations to survive. They're organic trophic. Okay, they include the yeast population. So those of you that are interested in fermentation, pickling, and beer making and wine, this is that population. Okay, they include molds. So they're strictly aerobic. These guys, they're filamentous often. Actually all the time, and they can extend really large distances, centimeters to, in some cases, meters and excess. Okay, the fungi, the largest organism. I always forget where this is. I want to say Washington State or something like that. Did I say this already? Did I mention this already? The largest organism in the world is a mold, or it's a fungus. Maybe it's Oregon, but it's out west someplace. Does anybody know where it is exactly? It's in Oregon. Yeah. Okay. The importance of these guys, there's a lot of interest right now in something that's called glomalin. Glomalin is a sort of a exudate. It's a part of. It's actually a lot of organisms do it. It's a it's a stress protein, but what it does in soils is it tends to create aggregates, and a lot of what we think of as granular structure, a lot of that's actually associated with the sort of the mycelia aggregates that are coming from the fungi. Okay, the mycelia are basically moving through the soil, and they're basically creating sort of aggregates, because it's basically sort of like a web, a root web. These mycelia spread out and they surround things. Okay, and then the proteins that are a part of this fungi mycelium, glomalin being one of them, basically stabilize this aggregate. Okay, and there's a lot of interest in this component of fungi. It's often also very much associated with plant roots. Okay, the next group is the eubacteria. These eubacteria are what we would think of, most of us would think of as bacteria, but there's another group we'll talk about next, which are archaebacteria, which are slightly different, but they're this sort of very small single-celled organism that are not necessarily aerobic. Okay, some of the important and really interesting kind of bacteria, I threw two of them out here because you guys are probably familiar with these even though you don't know it. The first one is actinomycetes. It's a class of bacteria that are very common in soils. They're rod-shaped as well as filamentous. These guys are, when you pick up a soil and you smell it and it smells healthy, this is actually the microorganism that's often associated with that smell. Okay, these guys tend to be fairly sensitive to toxicity and things like that. So when a soil gets degraded, these are some of the things that sort of disappear. Okay, Pseudomonas at the other end, there's lots and lots of different types of Pseudomonases. But the interesting thing about these guys is that some of these guys can use over 100 different organic compounds to survive. A lot of these guys, there's a lot of interest in these guys because of their ability to degrade things. A lot of remediation technology is sort of centered around some of these guys. This group also includes the nitrogen-fixing bacteria, which we've talked a lot about. These guys that convert the N2 basically to ammonium, but they prefer to use ammonium if available, and we've talked about why, because this is a very energetic process to convert that N2 to ammonium. Okay, so if ammonium is present, they're basically going to be using that. There's a whole group of different nitrogen-fixing bacteria, the rhizobia and the bradyrhizobia. Basically, these are the leguminous organisms that create those nodules in the legumes. There's a Zotobacter, which are basically free-living, and there's a Spirella. These are nitrogen fixers as well, and they have basically loose symbiotic relationships with some tropical crops, grasses, and grains. Okay, most of what we talk about around here are the rhizobium species, but there are a lot of other nitrogen-fixing organisms out there. All right, that brings us to archaebacteria. I apologize if this is going really fast, but I want to get to the next lecture, and this is more of a safari anyway. The archaebacteria are sort of broken up into three different large groups. One is the extreme halophiles. When you take halo, basically means salt. Okay? The methanogens and the extreme thermophiles, so extreme temperature. Of these guys, methanogens are really kind of interesting. They basically are methane producing, they're strictly anaerobic and they produce methane. Okay? They also can produce some other things, but they're basically autotrophic. They're basically taking CO2 and protons and basically making methane and water. Okay? So there's a number of other things that these methanogens are associated with: carbon monoxide, formate, methyl substances, and acetate. Okay? There's also these extreme thermophiles. These guys are really they're really important to microbiology right now. Does anybody know what PCR is? Polymerase chain reaction. It's a technique that's basically used to amplify proteins, DNA, RNA proteins. And the thought is, what you can do is you can take an organism, and if you want to find out if you want to explore this organism, well, this organism might not have a lot of the protein that you're interested in. Okay? And if it's at a very low concentration, it's going to make it very hard for you to discover things about it. You want to have a large amount of it so that you can do stuff. Okay? Well, it turns out that PCR is a technique that comes from, okay, well, why would I want to use PCR? Well, I want to be able to amplify it and make more of it so that I can take a look at it. Okay? Well, it turns out this technology came from a thermophilic microorganism, an archaebacteria, that was found basically in Yellowstone. Okay? Basically, you're taking an organism that was living in one of these geyser ponds, okay, extreme hot temperature, okay, and from this organism, they found this, basically I think it was basically a protein that, for whatever reason, amplifies other proteins, okay? And so they were basically using this organism to basically advance science in that sense. Okay, that's a very dumbed down version of the whole story, but it's actually a really interesting story. So, questions? Are we good? I know that was really fast. I apologize, but I wanted to get on to the next lecture. Go.
I have a quick question back on the methanogens. Those other compounds that you list, those are other things that they produce? So they can also, some of them can also produce formate, not consume?
No. I think these are things that they use.
They consume those
Yeah.
to make them.
Okay, questions? We're good? Good? All right.
This has been a production of Cornell University, on the web at cornell.edu.